we have sequencing data from 4 individuals, 2 from Colombia and 2 from Ivory Coast. These individuals have malaria, so they're infected with Plasmodium parasite, and we want to know in which of them Plasmodium parasite is resistant to one of the drugs, to pyrimethamine. So first, let's upload data from these individuals into Galaxy. So I'm opening Upload tool, clicking on Paste Fetch. This box appears, and I'm gonna paste URLs corresponding with the data to the data here. I'm gonna set this to fastqsanger.gzip. Check when you do this. It's fastqsanger.gzip, and I'm gonna click Start. This will put 8 data sets into my history. And now all data sets are uploaded. We have 8 data sets, although it's 4 samples. This is because this is paired end sequencing data. So for each individual, for example, this one ERR04222228, we have 2 files, one forward and one reverse. We can, of course, analyze these data sets the way they are. So we're just going to do the same thing over and over again 4 times because we have 4 samples. But we don't need to do that. We can bundle this data into a collection. And collection can have any number of data sets. 8 like here, or 100, or 1,000, or 10,000. So let's create a collection. First, we're going to click on this icon, which would make data sets selectable. We will select all of them. And for all 8 selected, we will build a list of dataset pairs. This will bring up this interface. So in this case, Galaxy is kind of confused. It needs to know which are the forward reads and which are reverse reads. So we'll give it a hint. Forward reads begin, well, they end with _F. You can see it filtered down to them. And these end with _R, and now it filtered down to those. And you can see that these correspond directly. So this one, ERR042228 forward, responds to ERR042228 reverse. So we can pair them. And so we can do for all the rest of them. So now Galaxy knows what the key hierarchy will be, how they will be organized inside this collection. And the last thing to do here is to name the collection. Let's call it data. And we'll click on this button. So the initial data sets disappear, well, they actually become hidden, and all we have here is just this one collection. We can explore it. If you click on it, you will see the samples. If you click on each sample, you will see the underlying data sets. So the first thing we want to do is we want to know how good these data are. And in general, raw sequencing data often comes contaminated with the sequencing adapters, which were not completely removed, and maybe it has bad fragments of the reads. So there is a tool called FastP, which looks through the reads, cleans them up, and also reports statistics on how good these reads are. So I find this tool by typing FastP in the search box. Here it is. And now I need to tell the tool that the data that we're dealing with here is organized as a paired collection. So I'm going to choose pair collection. And then it already knows what to select. But of course, you can always drag things around like this. We don't really need to change any other parameters here, so we can go ahead and run this tool. Now this is all done. And by the way, it makes sense to name your history somehow so you don't forget what it is. For example, something like this. Okay, so we just ran FastP. It generated 3 outputs. The first output is actual process FastQ files. So it's these data processed by FastP. So it's cleaned up FastQ. You can see that all of these things are collections because our input was a collection, then all outputs will also be a collection. So we can click on that and see our 4 samples, forward and reverse reads. These data sets are other artifacts generated by FastP. For example, it generates reports on how data with some quality statistics about these reads. And finally, it generates this log data in JSON format. JSON is JavaScript object notation. It's non-human readable format, but it's a machine readable format, and it can be used to visualize results of FastP. So, of course, we can look at quality of our reads by just going to HTML pages and clicking through all of them. But, if you have 10 of them, if you have 100 of them, that's a lot of work. We don't need to do that. We have a tool which can summarize data for all samples in just one go. This tool is called MultiQC. So let's find it. And this is the interface of MultiQC. It needs to know what results of which tool you want to visualize. So in our case, this was FastP, so we're going to choose that. And then we simply drag that JSON blob into here. That's all we need to do, and we run it. Now it's done. The output of it produces two outputs, but the one we really care about has this web page word in it. So if we click on this eye icon, we will see the graphical summary for all samples we analyzed. So one thing in this Galaxy interface, this is the tool panel. We can toggle it on and off. So I'll just remove it. Well, I'll hide it. So we have more space. So you can see that it gives us general overview of the data. For example, what's the GC content? Well, we know that Plasmodium parasite is very AT rich, so it's not surprising that these GC counts are low. There are no more adapters. There is some read duplication. And these datasets are not very large because, well, for this tutorial, I actually downsampled them, so everything runs faster. Perhaps one of the most important plots are these quality distributions. So this is the basis in the read, and these are the Fred quality scores. And in this case, so this line shows the two samples, they just overlay each other, so it looks like one line, but in fact it's a graph for both samples. And these two are the remaining two. And you can see that overall, of course, these are better than those, but in general, they're all good. They're all above 20, which is more or less where you want them to be. This is before filtering. And after filtering, you can see that it changes slightly, but not too much. Okay, so at this point, we can conclude that the data is of good quality, so we can go ahead and proceed with analysis. The next step of the analysis is mapping. So we need to map these reads against the reference genome. For this purpose, we are going to use a mapper, PWM-MEM2. But the mappers, they don't work by themselves. They take your reads and they compare them against an index. So it's a special data structure which is produced from the reference sequence, which allows mappers to find matches rapidly. In this case, we assume that Galaxy does not have the index. So we need to build the index and then map reads against it. In order to build the index, the first thing we need to do is to upload the reference genome. Let's do that. So we're going to go back to upload. And this is the old data that we uploaded. So I'm going to click reset. I will paste it here. Set this to fasta.gzip. And click start. Okay, it is uploaded. Let's go to tools and find PWM. PWM2. So here it is. Be careful, there are two of them. One of the indexer, just the thing that creates indices. We don't need this right now. And this one is the actual tool. So this is its interface. I'm going to hide the tools again. The first thing we need to tell Galaxy is whether an index for the genome that we're trying to compare against exists, or we want to use the one that we just uploaded. Of course, we want to use the one that we just uploaded. So I'm going to change this to use reference from history and build index if necessary. And this immediately will change this widget and will more or less automatically show us the only faster data set available in my history. This one can always make sure this by dragging, just to ensure stuff. You can always drag things. Our input data is paired collection. This one produced by FastB. So I'm gonna drag it in here. And we don't need to set any other options in this example. Let's click run. You can see what's happening here. Because we are using a collection, we interact with BWMM only once, but in fact, it runs 4 jobs. And because we have 4 samples. And if our collection had 100 samples, 1,000 samples, or 10,000 samples, so it would run 100 or 1,000 or 10,000 jobs. Mapping is done here. We can click on this collection and see that now we have 4 BAM data sets, because that's what mappers produce. Our goal is to ultimately covariance. So the next step is post-processing of BAM data, and one of the ways we post-process BAM data is we remove duplicates. And this is done with a tool called mark duplicates, this one. This tool needs to have BAM files as an input, so we're just going to drag this collection. And it has some options, and one of the options we're going to choose is if it finds the duplicate reads, let's remove them from the BAM file so they don't obscure what we do down the road. So just 2 things: tell it where the BAMs are and switch this to yes. Let's run it. Mark duplicates is done, and it just generates another set of BAM files without the duplicates. Now we are ready to go into actual variant calling. And so before we actually proceed with variant call, we need to do yet another post-processing of BAM files, and this is indo realignment. And this we will do with a tool called realign reads with low freq Viterbi algorithm. It takes the BAM files where the realignment needs to occur, and it asks where the reference is. So in our case, again, the reference is in our history, so we're going to change this from locally cached to history, and this will automatically find this because, again, that's the only FASTA file we have there. So with these 2 parameters, BAMs and the reference, we can run it. Realignment is done, and now let's use the actual variant caller. The variant caller is called low freq. And it's a variant caller which works well for haploid, viral, and bacterial samples. This is malaria parasite in human, and at that stage it is haploid. So this is the appropriate variant caller for this case, not GATK, for example. So again, it takes the BAM files. It needs a reference genome, so again, we're going to use one from history. And we need to make a few changes here. We want to call both SNPs and indels. And we want to set the minimum coverage to 10, so it's not going to look at the positions which have less than 10 reads covering them. Let's set the minimum base qualities to 20. Again, it's not going to look at any bases for reference or alternative alleles that have scores below 20. And let's set a mapping quality to 20 as well, which would pretty much eliminate any multi-mapping reads. So with these settings, we can click run. And now it's done. Let's look at what we have. So now we have 4 VCF files. And if we look at these files, so generally you can look at things by clicking the eye icon. But if you just click on eye icon, this will show things in the center pane. However, if you click this, the window manager, then it will open this in a separate window. And this way you can actually look at multiple things at the same time. Well, regardless, what we have here is a typical VCF file. So positions, what the reference allele, what's the alternative allele, and you can see that we have actually quite a few variants here. The problem with this is that you don't really know where they are. Yes, there are coordinates, but we don't know in which, are they falling within genes, are they in exons and introns and intergenic regions, things like that. So what we want to do is we want to add this information to this VCF file. And this is usually done with a tool called SNPF, because it essentially detects effects of SNPs. But SNPF needs a database. It needs to know where the genes are in order to annotate them inside in a VCF, like the one that like the one we see here. So we can create a database. In order to do that, we need to upload coordinates of genes. So again, click on upload, reset this thing, paste fetch, and set this to gtf.gzip. All right, so our GTF is uploaded. So let's go here and find the tool called snpf build. That's the tool which will build the database for us. So it needs to know the name for the database. Let's call it PF because we're working with Plasmodium falciparum. This is the GTF file we just uploaded, and the genome is in the history again. So this is our GTF file right here, and this is our FASTA file. We don't need to change anything else. Okay, done. And now we can use snpf proper, called snpf. It needs a VCF file with variants, so something that we generated with low freq. And it needs to know where to get the database. So in this case, database we just created. So we're gonna change this to custom snpf database in your history and drag this in here. We don't need to change anything else. Let's run it. Okay, so the output are also VCFs. Let's actually look at them. So let's look at this one. And let's compare it with the VCF which were originally produced by the variant caller. So we have like a side by side like this. So this is the original VCF. It only contains this information up to the info field. Well, the VCF, which was annotated with snpf, has considerably more information in the info field, such as, for example, it has the gene name. Well, in this case, it's an intergenic region, and it tells us this is intergenic region between which genes. For cases where it's inside the gene, and for example, indicates whether it's missense mutations, sense mutation, where it's introduced stop codon, all these sort of things. And it for amino acid, for mutations that cause amino acid replacements, it actually shows in which codon it occurs, what kind of change it is. So it really enriches VCF to a lot of additional information. Okay, so in principle, the VCFs can be visualized in the genome browser. But because we're using a custom genome here, we don't necessarily have a genome browser for this genome. So instead of doing this, let's format this variance as a table and then look for the markers of resistance for this particular drug. And we're going to do this with snp-sift tool called extract fields. And the fields we're going to extract are the following. Well, the chromosome position, what's the reference allele, what's the alternative allele, the one that we observe, quality, depth, alternative allele frequency, strand bias measure, counts for all reference and alternative alleles on both strands, the effect, the phenotypic impact, the gene where this variant falls in, and amino acid position, and as well as the codon change, well, change in the coordinates of the coding sequence and change in the coordinates of amino acid sequence relative to the beginning of the gene. We want to do one effect per gene, so in cases where you have, for example, two different mutations at a single site, they will be listed as separate columns. And we need to tell which VCF datasets we want SNP-CIF to extract fields from, which is going to be this one, the results of SNP-F. And we'll run it. Okay. If we look at the results, so we have still 4 files. And if we look at each, now you can see that this is a tab delimited file. So this is much easier to deal with. The other problem here is that we still have 4 data sets. So each sample is represented by its own set of variants. We need to combine them. But the problem with combining them, if we concatenate all these files, we are going to lose information to what sample they belong. Well, there's a tool for that. And if we go to collection operations and choose collapse collection and drag this in here, we can combine all the data sets. But when we're going to do this, we will also introduce a new column which will tell us which sample these data sets originally came from. Well, we want to keep the header, and we're going to prepend file name. So we're going to prepend essentially these ERR identifiers, which tell us which samples these are. Same line in each line in dataset. Yeah, it always surprises me. It doesn't make much sense, but that's what we need to do. Okay, let's see what we got. So this is now a single data set. And if we look at it, you'll see that we get this new column which tells us which sample it is. Still, there's just too much data here, and it's what, 1,500 lines. That's a lot to look at. So we know which gene is a potential marker for resistance. So let's just find lines in this file which have this gene ID in this gene column. And this is column 13. So let's use a tool called filter. Filter data on any column using a simple expression, and I'm just going to put this expression here. So column 13 equals the gene ID. Note that you need to put gene ID in the single quotes here. And we want to skip the header line because we need to keep something that would remind us what the meaning of the columns is. So let's run it. Well, this is it. Let's look at the results. So we have 5 lines. If you look at this carefully, you will see that, so we have one sample, we have another sample which is repeated 3 times, and we have a third sample. This particular mutation occurs in codon 108, so only 2 of those have codon 108 changed. And it's the same change from S to A, so it's a G to A mutation, G to A substitution, and it happens in sample 36028 and 42228. So these 2 are in fact resistant. The others, their 2 samples are not. So it looks like each location, in this case, have one sample which is resistant and the other sample which is not. We will also have another version of this tutorial which goes another analysis route. It uses a built-in genome reference from Plasmodium falciparum and uses browser to visualize these results.